Hello, my name is Sanjay Narsiwadir. I am a graduate student in the Computational Cognitive Neuroscience Lab at FIU in Miami, Florida. I will be presenting our work on a neurocomputational model of hippocampal field CA1 during context fear conditioning. The hippocampus is a cortical structure that is involved in a variety of learning and memory tasks. It is most vital, however, for performing tasks that involve rapid learning of complex stimuli. One such task is the contextual fear conditioning paradigm. Though hippocampal involvement in contextual fear conditioning tasks has been well documented, the precise mechanistic function of the hippocampus during these tasks remains elusive. Because subfield CA1 is the most important output structure of the hippocampus and is influenced by other memory-related areas, it might serve as a critical junction where contextual fear memories are stored and organized. Hence, we developed a neurocomputational model of field CA1 that takes into account the various inputs to the region. CA1 pyramidal cells are known to receive strong internal hippocampal inputs from neighboring CA3, relatively weaker inputs from layer 3 of the entorhinal cortex, and prefrontal inputs via the nucleus reunions of the thalamus. Based on experimental evidence, we assume distinct computational roles for each of these inputs to CA1. CA1 is assumed to generate a unitized memory in which various input representations are integrated to produce a single, sparse representation of an event. We assume that the EC3 input provides a distributed representation of events in which each stimulus or event recruits a relatively large portion of the given population, and different stimuli might recruit overlapping sets of neurons. The CA3 input, on the other hand, provides sparse and orthogonal representations, such that two different stimuli will recruit relatively distinct sets of neurons. While nucleus reunions is known to project to both excitatory and inhibitory cells in CA1, we focus on the inputs to inhibitory interneurons. We assume that the nucleus reunions-driven inhibitory inputs promote specificity of representation via a blanket inhibition mechanism. We simulated the spiking activity of each of these populations by constructing Izhikevich-style single neuron models whose parameters were fit to electrophysiological data. These models successfully reproduced the observed spiking dynamics of the cells from these regions. In order to quantify the influence of distinct inputs to CA1, we defined two related but distinct concepts. Sparsity refers to the manner in which neural activity is distributed across a population. A high sparsity score indicates that relatively few neurons are responding to the stimulus, whereas a low sparsity score indicates that most neurons are active in response to a stimulus. Similarity, on the other hand, compares the overlap in neural responses to two different stimuli. A low similarity score indicates that the two stimuli being compared activate mostly distinct sets of neurons in a given population. The similarity score represents a distance measure when inverted. Hence, neural similarity estimates obtained from our model can be transformed to behavioral response estimates by the application of Shepard's universal law of generalization. This law states that the response to any stimulus can be defined as an exponentially decaying function of the distance between the original and the new stimulus. Zhu and Sudoff in 2013 showed that projections to CA1 from the prefrontal cortex via the nucleus reunions were critical in facilitating discrimination between similar contexts in fear conditioning. In their first experiment, such projections were inactivated prior to the initial training session. During testing, inactivation produced increased responding to the similar test context in comparison to a control group, revealing an overgeneralization effect. In their second experiment, NR inactivation was performed before the testing session. Interestingly, the overgeneralization effect observed in the first experiment disappeared, revealing no meaningful behavioral differences between the control and NR silenced groups. Both of these results are accurately captured by our model. These pattern of results can be explained by isolating the computational effects of the various inputs to CA1 during learning. During training, the NR inputs increase inhibition in CA1, producing a sparse encoding of the contextual memory. When the animals are presented with a similar, overlapping input representation during testing, CA1 is able to encode a distinct memory that shows very little representational overlap with the training context. Once the original training context is encoded in a sparse manner, CA1 can activate a relatively distinct set of neurons during testing, even in the absence of inhibitory inputs from NR, as is the case in the second experiment. The sparsity of the original training context, established due to increased inhibition from NR, in combination with the orthogonal inputs from CA3 during testing, is sufficient to elicit effective behavioral discrimination of the testing context. In sum, our model suggests a clear computational mechanism based on blanket inhibition and pattern orthogonalization for the executive control of memory encoding via the nucleus reunions. Future work will include additional simulations of behavioral extinction as well as interference paradigms to shed light on the mechanisms by which competing memories could coexist in CA1.